on this somewhat humid Thursday afternoon. It is my great honor and pleasure to introduce today's speaker and, quite frankly, one of my science heroes, Dr. Kent Berridge, who is the James Olds Distinguished University Professor of Psychology and Neuroscience at the University of Michigan. So Kent received his Bachelor's of Science in Psychology at the University of California, Davis, and then went on to do his PhD at the University of Pennsylvania. And then after a 2-year stint in Canada at Dalhousie University, he took up his faculty position at the University of Michigan in 1985 and hasn't looked back since. So Kent has had an illustrious career that is focused on clarifying the neurobiological mechanisms that underlie emotions, motivation, learning, and reward processes, and how dysfunction in these systems may relate to different psychiatric disorders, including drug addiction, depression, and other syndromes that are associated with deficits in these types of things. Now, his research has been tremendously influential, and probably he's best known for his work that has, in exquisite detail, clarified what role dopamine does and does not play in processes related to reward. So you've all heard of dopamine, and probably all have heard that it has something to do with reward. But this idea has been kind of conflated and misconstrued a bit so that dopamine might equal pleasure. And so indeed in popular media reports you hear, this increases dopamine, the brain's pleasure molecule. Well, those of us who study the dopamine a little more seriously kind of know, due in no small part to Kent's seminal work, that this is not quite right. And in fact, just because brain levels of dopamine may increase when you're biting into that really good slice of pizza or snuggling up to your significant other, it's not that dopamine that makes you like this experience. It's not mediating the pleasurable or hedonic sensations associated with that. But rather, it may be more involved in the motivational aspects, the wanting, the craving to get to those. So dopamine helps you get to those good things in life, but doesn't necessarily make you like them when you consume them. And other systems that I'm sure Kent is going to talk about today, dissociable systems, may be more involved in the hedonic, pleasurable aspects of reward. So he has had a prolific career, regardless of what metric you want to choose. He's 150 plus publications, an H-index between 65 and 80, depending on which website you want to believe. He's been cited by the faculty F1000 numerous times. He's won numerous awards. He was a Fulbright scholar. He's a charter and fellow of the American Psychonomic Society, the AAS, the APA. In 1991, he won the APA's Early Career Award for distinguished contributions. And then just to prove that he wasn't a flash in the pan, 25 years later, he won the APA's Senior Distinguished Scholar Award as well. So he has been showing research excellence for his entire career, something we can all aspire to. Now, I could literally go on for another 20 minutes, but I promised Kent I would let him talk a little bit. So if you'll join me in welcoming Dr. Kent Berridge.
Thank you, Stan. That was certainly more kind than I deserve. And it's a real privilege and pleasure to come here to UBC. It's been one of the top departments in psychology and one of the top universities in neuroscience for decades. Many of my favorite papers over the years that I've read have come out of the labs here at UBC, and especially in my own favorite topic of the behavioral neuroscience of reward and motivation. This place has always been a leader, so it's a great pleasure to come. Today I want to talk about aspects of reward, and we will talk a little bit about dopamine too. Let's start with the notion that didn't have to be true, a notion that I didn't believe even 15 years ago. It's been almost surprising how many human psychological, cognitive, and cultural rewards have turned out to share the same brain mechanisms as sensory pleasures of drugs and sex and food that we've known about in years for animals. About 15 years ago, I was involved in a human neuroimaging study where we thought that food pleasures in people would activate subcortical nucleus accumbens, dopamine-related systems, and that art and humor and music would activate cortical systems, that these would be separate systems for pleasure, cortical cognitive culture and subcortical sensory pleasures. We were wrong, and 15 years of much better neuroscience imaging studies have shown that we were wrong. That sex and drugs in humans activate the subcortical systems as we'd thought, but also, as studies in Canada at McGill University have shown, favorite music. For music students, listening to the music that gives them chills of pleasure activates the same systems of music, of subcortical as well as cortical systems, and even sustained well-being, a general feeling of happiness rated in people, this by Richie Davidson at the University of Wisconsin, correlates with nucleus accumbens activation. In short, it seems that it's the same usual culprit, the same brain systems for reward that are recruited in all kinds of rewards, from music and well-being down to food and drugs. And what this means is that what we could learn about reward from studying one kind of reward may tell us something about brain systems for all other kinds of rewards in people too. They feel different, but they share something in common. Now, reward is a word, but the psychological processes of reward can be divided into lots of different subcategories. And this is a diagram from a paper with Terry Robinson, my colleague at the University of Michigan, that kind of breaks up reward into various kinds of cognitive and cue-triggered kinds of wanting and conscious pleasures versus core, less consciously acceptable hedonic liking reactions. We can simplify it all, though. We don't have to deal with a real complicated roadmap into just these three separate categories. Virtually all rewards involve an aspect of something we like, we want, and usually we've learned about them. Most rewards we've learned about, or we expect them based on what we've learned in order to expect and to imagine. So what's doing what in the brain for the psychological processes of reward, for the psychological components? Well, let's start with dopamine. It's just a random choice in a sense to choose dopamine because dopamine is only one domino in a chain of dominoes in the reward circuitry. It's a domino that's particularly easy to manipulate with drugs and lesions and optogenetic stimulation. It's a domino that's particularly consequential and powerful in its effects on rewards. And it's a domino that is relatively easy to measure with electrophysiology because the neurons cluster in one place, or to visualize dopamine projections because they pass through one, or to measure release in their target structures with microdialysis or voltammetry or radioactively labeled PET studies in people. But it's just one domino in the chain. Still, historically, it's been such an important domino that it's worth starting with it. And I speak as one who actually did believe that dopamine was pleasure. I believed it very strongly because I was persuaded in the 1980s up to about 1990 by the very persuasive and elegant work of this fellow Roy Wise, who was then at Concordia University in Montreal. Roy was famous for his anhedonia hypothesis of what happened if you suppressed dopamine. He said you blocked all dopamine pleasures because his hypothesis was that dopamine junctions really did represent a synaptic way station where sensory inputs are translated into the hedonic messages we experience as pleasure or yumminess. Now, I believed he had elegant data to show that this was true in all kinds of tests for food reward, sex reward, drug reward, brain stimulation reward. If you suppress the dopamine systems, the animals wouldn't work for those rewards. It was as though the value drained away, and he had clever ways to show that they weren't just getting tired, they weren't just feeling suppressed, they were specific to that situation, they could bounce up again. Roy actually phoned me in the late 1980s and invited me to visit him in Montreal. So we decided we'd do a collaboration. He sent down a graduate student that following Christmas vacation to Ann Arbor, and the graduate student, Louis Coley, gave dopamine-suppressing drugs to rats, and we looked at a way to kind of measure yumminess because we were going to actually test this hypothesis that dopamine would experience as pleasure yumminess. But our results didn't support that. How would you measure yumminess? Well, in this collaboration, we were doing taste reactivity studies using a measure of yumminess that human parents have used to ask their infants for eons whether the child likes this particular food that the parents are eating, and that Jacob Steiner had developed into a test we'll see in a second, and which Harvey Grill and Ralph Norgren applied to rats. These liking reactions to tastes are modulated by hunger and appetites and satieties. They're modulated by psychological preferences and aversions and by brain stimulations. Here's what they look like. This is from Jacob Steiner in Israel. An infant, that infant just got, let's see it again, just got a taste of sugar water on its tongue through a dropper. And what we see is sort of relaxed face and tongue protrusions. Here's a rat getting sugar water infused into its mouth painlessly through oral cannula, painlessly. We're looking at it from underneath. And we see these tongue protrusions. We slow it down so we can see the tongue protrusions to the sweetness, these licking of the lips, lateral tongue protrusions, licking to the sweetness. Bitterness is different. Gapes and sort of scrunching of the nose and the eyes. Rats can't scrunch their nose and eyes. They don't have the musculature, but they can gape and shake their head and flail their paws and rub their chin on the ground. That's what they'll do to bitterness. It's not just sweetness versus bitterness. As we said, we could take sweetness and make it elicit this gape response if it had been paired with visceral illness, with nausea, to create a learned taste aversion. We could increase the liking. We can make gapes to some things become liking, as we'll see in a moment, if we, say, make intense seawater saltiness suddenly positively liked through a salt appetite, a physiological salt appetite. So it's not sweetness versus bitterness so much, it's whether the taste is liked or not. And we all expected in the late 1980s that giving dopamine blocking drugs would suppress the liking reactions to sugar and maybe even increase the bitter gaping responses. But nothing happened at all. Nothing happened to these facial reactions, and we didn't really think much of it. It was our first dopamine experiment. It was Roy's graduate student's first taste reactivity experiment. We repeated it a couple more times and got the same results. I had a colleague, and I thought, well, maybe drugs were just not strong enough. My colleague Terry Robinson at the time was doing dopamine lesions, creating Parkinson's-like rats on one side of the brain with a dopamine lesion on one side of the brain. And we thought, why couldn't we do that on both sides of the brain to simply take out the dopamine system, eliminate 99% of it, a neurochemically specific dopamine lesion that would leave them sort of Parkinsonian. After that lesion, the rats never ate, they never drank again, they showed total aphasia and adipsia. We would maintain them through intensive nursing like we'd all receive in a hospital with intragastric feedings 3 times a day and keeping them in good health. We gave them the tastes. Here's what they look like. This is getting sugar responses to the taste, and we can see here 2 dots. These are, the black dot is the hedonic liking reactions to sugar of normal rats. The white dot, almost next right part of it, is the response of the dopamine lesioned rats who've lost 99% of their dopamine, more than 98%. Not really a difference, and neither of them is at all aversive. We wondered, well, were they still expressing palatability liking? Could they, say, change their liking to a sweetness if we created a taste aversion to it? Or were they now just reflexive? So we gave them a learned taste aversion to a sugary taste. This is a response to the same taste after they'd learned the aversion. And you can see both groups, they dropped all the liking reactions, and now they're showing gapes and things to the sweetness as though it were bitter. Perfectly normal liking, perfectly normal learning of new likes and dislikes these dopamine-free rats could have. And that was a disappointment. We had to make sense of it, and it led us to the incentive salience hypothesis. But for 10 years, it was a lonely desert of having to say that we didn't think dopamine was pleasure because so many colleagues knew that dopamine was pleasure, or at least they thought they did. There was beautiful work here by Tony Phillips in the early 1990s showing that dopamine was really linked more to the incentive motivation value of things than to pleasure. But Tony was one of the only, I think, people to agree with us in the 1990s. It took another 10 years before human studies started to be done that would support the notion that maybe you could separate liking from dopamine. One of the first was done by a former colleague of Roy Wise's, Harriet DeWitt, and her graduate student, Lisa Brower. Harriet was an author on the anhedonia hypothesis, so she believed dopamine was pleasure, certainly. And she was going to suppress dopamine systems by giving people pimozide at the same time that they were taking amphetamine. And the notion was suppressing the dopamine should reduce their liking for the amphetamine. And what they found was that it did reduce their ratings of feeling stimulated, but it didn't reduce their ratings of liking the amphetamine. She did it a first time and got this result, and then she did it a second time. This is the second paper that they published together with a higher dose of pimozide, and it still didn't suppress it. You can see this is the amphetamine, this is the 0, this is amphetamine, higher dose getting liked, and then if you add pimozide to that, these bars are basically equivalent across. The pimozide just wasn't suppressing the liking ratings of them. That was the first, and it was followed soon after by people like Marco Layton at McGill University, who suppressed dopamine in a different way by giving an amino acid cocktail mixture of amino acids that compete with dopamine synthesis, so dopamine synthesis is suppressed for a few hours after drinking the cocktail. He gave people cocaine at the same time that this was happening, and here we're seeing the effects on their ratings of wanting cocaine, wanting to take more cocaine, versus liking, euphoric liking for the cocaine. And basically, the blue and red lines, we are seeing a suppression of the ratings of wanting to take cocaine, especially for the highest doses of cocaine. But there's really no suppression of the liking ratings. They're staying pretty much intact. The world began to change its mind, I think, with these kinds of human studies. The difference between liking and wanting for dopamine, stepping back a decade again to the early 90s, had led Terry Robinson and I to propose this incentive sensitization hypothesis of addiction. It was really Terry's idea to do this, and it was based upon partly the notion that dopamine was wanting, but also the notion that he was discovering with Jill Becker and others in his lab and in that field, that drugs of abuse like cocaine and amphetamine and heroin and morphine, they don't just activate dopamine systems. They do activate dopamine systems, but they don't just activate it while you're taking it. If you take it in repeated doses, especially in binge-like intervals with pauses in between, like taking it at high doses on the weekends and not during the week, and especially for some individuals who are vulnerable to this, you develop a semi-permanent change of neural sensitization of those dopamine systems, where they become not hyperactive, but hyperreactive. You can tap into them at any moment and they look fairly normal until you give them the right stimulus, and then they're hyperreactive. The right stimulus could be the drug again, an addictive drug, which elicits more dopamine release and many hyperreactivity changes in the circuit, both before dopamine and after dopamine in neurons in that circuit. And once it's induced, this neural sensitization, it can be induced in the same individual who's developing tolerance if they're taking dopamine, the drug, again and again and again. Tolerance is due to downregulation of receptors and things, and it lasts as long as you're taking the drug, but tolerance tends to go away if you stop taking the drug. Many of those compensations come back. Sensitization doesn't go away when you stop taking the drugs. It may even incubate in a time of no drug taking, a thing called incubation of craving, and actually emerge even stronger. And it's tapped into by the drugs again, and tapped into by cues that are associated with drugs. Cues are going to be very important in the rest of this talk. Incentive sensitization was really just posed for drug addiction, for people who take addictive drugs. Not everybody's equally vulnerable. Some are resistant. It depends on genes. It depends on things like sexual steroid hormones. There are sex differences. It depends on previous experiences, like prior traumatic experiences that can cross-sensitize to drugs. But it's invented just for drugs. In the last 10 years, though, it's begun to expand in its applications. So here is sort of a test case, a kind of laboratory experiment that no one would ever do ethically, but one stumbles into doing with people clinically with Parkinson's patients. That's really been induced by the invention of the new medications for Parkinson's disease. Parkinson's patients are the sort of least likely to be addicts that one could think of. They're not known to be sensation seekers, but with the new medications that actually tap into receptors for dopamine neurons, the old medications would cause remaining neurons to synthesize more natural dopamine and release it. The new medications jump past dopamine itself to key right into the receptors and stimulate the receptors. In about 15% of the patients who take these direct dopamine-stimulating agonist medications develop a number of behavioral addictions like compulsive gambling, compulsive sex, pornography pursuit, compulsive shopping, sometimes compulsive binge eating, compulsive punding, things like sorting buttons or engaging in hobbies. And often a patient who shows one of these things may show another one or two, usually several of them together. What's happening to create this? Well, I don't study these patients, but others who have given them things like L-Dopa and found that these patients who are developing it are actually showing a heightened dopamine response. It's the blue bars to their medications. So they're actually giving a higher dopamine response. And these authors, O'Sullivan and colleagues, are suggesting that the medications has essentially reached into Parkinson's patients and created them from least likely to be an addict into an addict, a behavioral addict with these kinds of behavioral addictions, so that they are showing a sensitized signature to these things. It's not just behavioral addictions they can show. Sometimes these patients actually become addicted to their medications. Some of them demand far too much L-DOPA medication, going from neurologist to neurologist to neurologist to get more prescriptions. L-DOPA is not an abused drug. It's not sold on the street. And Marco Layton has given it to normal people, and they don't report it's particularly pleasant. But these patients are pursuing high doses. It was suggested for a while that they were pursuing these high doses because they were going into withdrawal in between the high doses, so they were pursuing the drugs to come out of withdrawal. But that seems not to be true. Here, they're actually giving the drug, and you can see the brain dopamine release correlated with ratings of wanting to take more drug when they've just gotten an initial dose, versus liking, and you can see there's not a correlation of the dopamine release to the liking. These patients who are on their medication have good motor function and good ratings and normal ratings of drug liking, but they have greater ratings of drug wanting despite even actually developing dyskinesias when they're under the medication. So the L-DOPA challenge is actually inducing peaks of drug wanting. It's not eliminating that craving that they had. Dopamine stimulation can drive this kind of wanting, and Parkinson's patients are not anything one would never wish to create this deliberately, but it's sort of a test case that's possibly the best human evidence that reaching into the brain with dopamine stimulation can turn on addictive-like wanting, even in individuals who aren't otherwise inclined to show that. In the last couple of years, behavioral addictions like gambling and sex pursuit and various kinds of compulsive behaviors are emerging that are also perhaps fitting this kind of brain signature pattern that some authors are suggesting is sort of fitting into cue-triggered hyperreactivity in these particular individuals, as though spontaneously some of us could actually become sensitized even without taking drugs. It's actually been known for decades that it is possible to sensitize without taking drugs. It was known that traumatic stress would sensitize. Things like a strong salt appetite induction would sensitize. Eileen Bernstein at the University of Washington had shown. It may be that pursuing some of these rewards in some of us is inducing sensitization that could be contributing to some of these compulsions. Again, this is a controversial hypothesis, and it isn't something that I'm advocating. It's being tested by other people, so it will remain to be seen how there are people here who can evaluate whether this is going to be the right story. The final kind of application to human clinical situations of this wanting versus liking distinction has also emerged in the last 10 years by people like David Zald and Michael Treadway and Deanna Barsh, thinking about clinical anhedonia syndromes in diseases like Parkinson's disease and schizophrenia and major depressive disorder. Patients in those conditions have long been considered anhedonic. The textbooks say that they're anhedonic going back several decades. It's as though rewards have lost their value for them. But studies that have been probing it with specific sensory pleasures find that actually they have normal liking and capacity for pleasure. It's just that they don't value any of these rewards anymore. So the term anhedonia is being suggested by some to be replaced with something like avolition, or a loss of motivation, amotivation, rather than actual loss of pleasure. And so it's a reinterpretation of anhedonia as not actually a loss of pleasure after all. The very last thing before I leave dopamine, the very last kind of application is sort of the dark side of dopamine. Dopamine's famous as a reward neurotransmitter, but it may also have the capacity to participate in certain forms of fearful motivation too. This has been pointed to in things like the dopamine hypothesis of schizophrenia, to the degree that schizophrenics may have hyperreactivity in subcortical dopamine systems while having maybe suppression in cortical dopamine systems. It's been suggested by people like Satish Kapoor, who was at the University of Toronto and now is at University College London, and his colleagues, that extra dopamine in subcortical systems is responsible for generating sort of the paranoia aspects of percepts and stimuli in the world. We can all step outside and see a police car and perhaps feel a little bit comforted and safe. But for somebody in this sort of state who sees a police car, it may have a special meaningfulness, which is almost like incentive salience. With incentive salience, you can't not look at what it is that has this special motivational salience. But unlike being attractive, something with this fearful salience can be threatening and make it almost repulsive and sort of call forth a kind of fearful active coping response. In the lab, we can produce a dopamine involvement in fearful coping responses by doing things like this. This is looking at the nucleus accumbens from the side, and if we blow it up here, you're seeing a color map. It's not a neuroimaging map, it's a map of causation. Every little splotch here is a micro-injection of a drug that's going to inhibit nucleus accumbens neurons and release targets downstream in other brain structures from constant nucleus accumbens inhibition. So it's going to activate targets through this sort of way. It's the same drug in all these colors. Wherever you see green, it's sort of turning on wanting. It's making rats eat more. It's making them return to a place that this microinjection was paired with. It's basically an incentive wanting-like response wherever it's green. But the same microinjection in the back of the nucleus accumbens is eliciting fearful-like responses. If you try to touch the rats, they squeak and try to jump out and bite you, but normally our rats are very tame, but after these red microinjections, they'll try to bite you. They'll also show defensive burying, which is a response that was studied here at UBC by John Pinnell and Dallas Treat many years ago, where rats will sort of will bury a threatening thing that gives them an electric stimulation, the rat electric shock. The rats will, in this case, where you see red microinjections, they'll defensively bury towards us in the room as though they're afraid of us. You can see green versus red, and both of these effects require dopamine. They both require dopamine, and the entire nucleus accumbens can flip back and forth to some extent between a desire-generating system and a dread-generating system. Sheila Reynolds and Jocelyn Richards showed this in our lab by doing things like making these same micro-injections that you're seeing here, but just in different situations, like, say, here. You probably can't hear that. There's not much sound there, but that's Iggy Pop, who was actually a University of Michigan student before he dropped out and pursued some other career. If you play this to rats, this music, and turn up the lights, they don't like Iggy Pop. They'll avoid a place and they'll turn him off. This is the first 15 minutes of a 1-hour test. The black bar is Iggy Pop. They've turned him off. By the end of the hour of the 1-hour test, he's completely off. So they'll choose to turn him off. But what if they can't turn him off? What if you just do the microinjection and then place a rat in a situation where Iggy Pop is playing? What happens is that the red area expands. The yellow spots are spots where both desire-eating behaviors and fearful behaviors are being elicited from the same rat in the same test. So both desire and dread. It sort of expands so that only a little front rim of the nucleus accumbens remains pure desire generating. If we move beyond this, if we switch the rats back to their homeroom, which is dark and quiet and smells like rats, the entire nucleus accumbens blossoms into a pure, almost pure desire-generating zone. All of these effects require dopamine. If we block dopamine, all the colors fade. There's no motivation of either kind. The dopamine use flips a little bit when we flip from green to red. It flips from involving just D1 dopamine receptors to requiring also D2 dopamine receptors. The neurons are actually switching their neurobiological modes. But it's a dopamine-dependent generation, and you can see how it might be related to things like a paranoia, a fearful kind of salience there. Okay. So all of that is about wanting. We started looking for dopamine as pleasure liking. It didn't turn out to be that way. Wanting is something separate, and we'll come back to wanting again in a minute or two. But what about liking? For years, when we were saying that dopamine wasn't wanting, colleagues would say, so well, then what in the brain is real pleasure liking? It turns out we can turn on pleasure liking in the brain, never with dopamine in our hands, but there's several other things that will do it. Here we're seeing this representation of the brain with dopamine is green for desire. There's a large, robust network in the brain for generating desire, and you've just seen a little bit of it. But liking are these little red spots that we can see here. We call them hedonic hotspots. They're nestled within each of the structures that contains them, usually very tiny hotspots, and they can be turned on neurobiologically with neurotransmitters, but never dopamine. Instead, things like opioid or endocannabinoid or related neurotransmitters. Here's a rat brain blown up, and we can see from the side 2 hedonic hotspots. To turn on intense liking, we need to stimulate at least one of these hotspots, but it's a fragile circuit, and it's all integrated together. If we stimulate one of these hotspots, it will recruit the other, but it requires that unanimous recruitment of all these hedonic hotspots to get a pleasure liking enhancement. And if we disrupt that unanimity by turning on one while blocking another, we won't get enhanced pleasure liking. There's hotspots in the prefrontal cortex, in the nucleus accumbens, the ventral pallidum, and even in the brainstem. To just give you an example of one, we can look at sort of the first one we discovered. This is a close-up look, again, at the nucleus accumbens. It's just like before, the nucleus accumbens sideways, sagittal view blown up sideways. And wherever you see a color splotch, again, it's a causation map. These are microinjections of an opioid-stimulating drug, Damgo, to stimulate mu-opioid receptors. And wherever it's green, the size of the microinjection splotch we can detect by measuring Foss plumes of neuronal activation, but the color of the plume tells us what it's doing psychologically and behaviorally. Wherever it's green, the rats are eating twice as much as usual, or 4 times more than usual, or 8 times more than usual of things like sweet M&Ms and chocolate candies. And the green stretches under the red, and the green would come out into the room and go further behind the screen. The green would fill the entire nucleus accumbens and extend to parts of the neostriatum and up into the parts of the amygdala. The green is robust. The red are places where not only is it increasing the wanting to eat, but it's actually increasing these liking reactions to sweetness when the rat gets them. And there's no rational reason why those little red and orange spots should be clustered together like they are, but they are. This is what defines the hedonic hotspot. This hedonic hotspot, it's about 1/3 of the medial shell of nucleus accumbens that we can see here. It'd be about 1/10 of the entire nucleus accumbens coming out into the room too. Just 1/10 of the nucleus accumbens. That's where we can double or triple these liking reactions as well as increasing the wanting. That was from a study 10 years ago, but it was recently replicated here by Daniel Castro. And basically, he's just showing us again this hedonic hotspot in the rostral part, and also an opposite-coded hedonic cold spot, blue, in the back of the nucleus accumbens, where opioid stimulation actually suppresses liking, even though it still increases wanting to eat as much as in the hotspot. So even opioids can behave like dopamine, even opioids can create wanting without liking, even opioids can create wanting with suppressed liking if you put them in the proper spot. And the same general anatomical rule applies to other opioid receptors too. I used to think up until about 3 years ago that all these hotspots would be subcortical. All the ones we had found up until 3 years ago were subcortical nucleus accumbens, ventral pallidum, brainstem. But Daniel Castro recently in our lab started mapping limbic areas of cortex by making microinjections wherever you see these splotches. And here's what happened for hedonic enhancements. He's found a hedonic hotspot in the orbital frontal cortex. Here we're seeing a sideways view of the brain, the cortex from the outside, the lateral surface. And here we're seeing the midline, right along the midline, the cingulate surface. And this collection of red here is actually joined together. It's just going laterally to the front and then crossing to the middle. So there's a medial orbital frontal cortex hedonic hotspot where opioid or orexin stimulation increases liking reactions. There's another hedonic hotspot in the posterior insula, far, far back in the insula. And then there's sort of a suppressive cold spot where the same neurochemical stimulations suppress liking. So the cortex is controlling liking. It's contributing it to it in this sort of hierarchical way. All of those hot spots are places where we can double or triple or quadruple the intensity of the liking reactions. Each time we stimulate one of them, it will recruit Foss not only where we stimulate it, but also in the other hotspots too. So it turns on the network as a functional network whenever we enhance liking. And as I say, if we turn on a hotspot but we stop the rest of the network from activating, there'll be no hedonic enhancement. You need this unanimous recruitment of all the hotspots together as an integrated network in order to create intense liking. That's relatively difficult to do compared to generating intense wanting. That's so easy to do. And this may be a reason why intense pleasures in our life are relatively few and far between compared to intense desires. There's a difference in the circuitry and in the ease of activating that circuitry. The last thing I should say about these hotspots before I leave this liking topic is that we might well expect that if something's causing a pleasure enhancement, then taking it away would cause a pleasure reduction. But it turns out that that's not always the case. The brain is arranged hierarchically. In fact, there's only one spot in the brain, one spot in the brain which, if taken away, nice things like sweetness will become nasty and disgusting. A tiny spot in the brain. And my jaw almost drops whenever I hear myself saying this, because how could it be that there's only one spot in the brain which, if taken away, nice things become nasty? That spot is the ventral pallidum. It's a hedonic hotspot in the ventral pallidum. It's a hedonic hotspot where opioid stimulation will triple or quadruple liking, just like you saw in the nucleus accumbens. But it's the only spot where a small lesion will make sugar be responded to as though it were bitter and disgusting. For this to be true, it's telling us something important about the affective wiring of brain systems. And I don't think we can fully wrap our minds around just exactly how it is true of the implications of it. But I think that's an important thing to know about affect in the brain, pleasure in the brain, that only this one site will destroy liking and make nice things be responded to as disgusting. If you know of any other sites, please tell me. I'd like to focus for the rest of my talk really on the relationship of wanting to learning and to try to kind of pull apart desire and show it to you a little bit more clearly, both in its psychological properties and in its neural bases. Wanting is usually for things we like, but we've seen we can pull liking and wanting apart. Wanting is usually for things we've learned to want, learned to like, or we've learned about and we have rational reason to expect that we're going to like based upon learning. So let's look at the relation of wanting and learning. We're going to call upon key features of incentive salience, psychological features, in which Pavlovian cues for reward become extremely important, not just in informing us about what to do, but actually in turning on motivation. If anybody here recognizes the name Dalbor Bindra, this is where this notion comes from. He was a psychologist at McGill University. Pavlovian cues become able to trigger motivation and reward. They can trigger phasic spurts of motivation. In humans, vivid imagery about rewards can do the same thing as Pavlovian cues. In animal experiments, we have to do Pavlovian cues. But the thing about Pavlovian cues is that they're not always motivating. The same cue can be intensely motivating on this occasion, somewhat on this occasion, and not at all on this occasion. Our brain is modulating the motivational power of the motivating capacity of learned cues. So wanting is going to decouple sometimes from learned reward predictions. This is going to be adaptive, but it's also going to set the stage for truly irrational desires that we'll see in a minute. Okay. Maybe to just give you a sense of how this works, can you imagine ever wanting something that you know from past experiences is truly disgusting? You've had it many times and it's always been disgusting, and it's only ever been disgusting. Can you imagine yourself now wanting that intensely? There you go. Yes, it's very possible. We can create it in the laboratory too. Let's see. Here's a case to do it, but just to kind of see how we're going to do it, let's first see how do we normally think about learning about rewards interacting with what we want. Anybody who studies learning in sort of associative frameworks, Rescorla-Wagner frameworks, Pavlovian conditioning, temporal difference, prediction error, equations that make model-free cases, it's all the same. It's incremental learning. Incremental learning is a case where we learn a value slowly by repeated trials, and then supposing the value suddenly changes. How could it change? Well, if for a particular reward, we might create a new satiety state that would make it less valuable, or a new hunger state that we've never been in that would make this reward more valuable. But we haven't yet experienced the reward in that state. So what's going to happen? Well, many experiments have shown that in this kind of situation, what happens is you start wherever you left off, and you gradually, incrementally work your way up to a new higher value if you've got an appetite state, or you gradually work your way to the lower level if you've got a satiety state. That's associative learning by experience. You start where you left off. Now, it's possible to more dramatically change value in a cognitive way. We are cognitive creatures, and even rats are cognitive creatures. So if we've learned something has a particular value in a cognitive state, some of you will be familiar with Tony Dickinson and Bernard Berlin's animal work on this, but we can all identify with as a person. In the animal experiments, Tony Dickinson and Bernard Berlin will train a rat up to a particular value of a reward, and then they'll create a new state, and then they'll pop the rat into an extinction test. An extinction test where the rat can go back and work for that reward, but there's no reward today. It's extinction. It's like extinction mimicry with the old dopamine suppression. Extinction, no reward. If there's no reward, the rat starts out working, and it just gradually falls down. It's just pure extinction. This is normal extinction if the rat isn't in the same state as it was trained in. If it was in a hungry or sated state, it looks exactly the same. That's the point of the Dickinson-Berlin extinction experiments. But you can instantly change the rat's value. All you have to do is give it one retasting outcome of the reward, the old reward in the new state where it has a higher value, or the old reward in the new state where it has a lower value. Just give it one retasting. It could be at home. It could be in another building. One retasting where you realize in this state, this is a marvelous reward, or this is a terrible reward, and now you put the rat into this test situation, and it instantly pops up to the new shifted value. So we learn cognitively by this sort of epiphany by experience. But it's still an epiphany by experience. We have to retaste. That's the crucial thing about these cognitive outcome things. Okay, so suppose now we learn that something's truly, truly disgusting. Like, well, salt. We all like salt. We like salt, pour it on food. But if you've ever been to the beach in the ocean and you swallowed a mouthful of seawater, a big mouthful of seawater is not so nice. If you've ever been to the Dead Sea in the Middle East, which is 3 times saltier than seawater, and swallow a mouthful of Dead Sea seawater, that would be truly, truly disgusting. Here's a rat who's going to learn that a particular cue predicts Dead Sea seawater squirts into its mouth. And it's terrible, terrible, disgusting. And then one day it wakes up in a state that it's never been in. Here's the situation. The rat's going to have actually 2 cues and 2 rewards. It's going to have a situation where a lever pops out of the wall, and the lever is just a cue, a Pavlovian cue. When the lever pops out, a few seconds later, a sugar squirt will come into its mouth painlessly through oral cannula. And you'll see that it becomes attracted to the cue. A different lever will pop out of a different wall, and this lever will predict Dead Sea saltiness. And you'll see the rat kind of avoids this salty lever, right? Now, one day, suddenly, the rat wakes up in a state that it has never been in, that you and I have never been in, but our ancestors were in, and there are squirrels and deer outside now that are in. It's a state of sodium deficiency or salt appetite. Sodium deficiency develops if we have not been eating salts, if we've been, say, purely vegetarian and haven't had any salt supplements, we'd be in sodium deficiency. But we can induce salt appetite overnight by just giving hormones that mimic the signals to the brain that normally signal sodium deficiency. Aldosterone to stimulate receptors in the extended amygdala and brainstem, coupled with angiotensin II that will trigger receptors in the forebrain and hypothalamus. These 2 combined signals create a state of salt appetite. On this day, even Dead Sea seawater would be liked. But the rat's not going to get the taste of Dead Sea seawater until just a little later in the day. Our question is, what will it do to the cue that predicted terrible, terrible, disgusting seawater? Does it have to retaste in order to want this cue? Let's see. You're going to see the rat first on a training day, and then in the sodium deficiency day. So here's a training day. The lever pops out of the wall. This is the sugar lever. You can see the rat's very eagerly on it. And then this is just protecting the tube that's now delivering the sugar. Now the sugar's coming in, and the rat's showing all the liking reactions that you had seen earlier on. Here's a different lever over here. This lever pops out, and the rat stays away. This will predict the Dead Sea saltiness. The rat stays away, stays away. That's a normal training day in normal state. Now this is the day in the new state. The rat wakes up in the new salt appetite state, and this is the very first lever on the very first day. The rat has never tasted good salt yet as good, and you can see it's on the salt lever as eagerly as it had been on the sugar lever, even though it has never, ever experienced salt yet as anything but disgusting. Later today, it'll taste it, it'll be nice, but it has never been nice in its life. How is this happening? How is this happening? The answer is very simple, neurobiologically and fairly simple psychologically too. Neurobiologically, what's happening is that when the rat encounters this salty lever in this state, something happens in its brain that has never happened before, and that is activation of the mesolimbic dopamine system and its targets. Dopamine neurons in the ventral tegmentum are suddenly activating with phos, and targets in the nucleus accumbens and in the prefrontal limbic areas of the cortex are activating, all activating. It's not salt appetite that's doing this. If the rat's getting salt appetite without that salty lever cue, this doesn't happen. It's the combination of lever plus cue in the state creates this new value. What's happening psychologically is that the rat had learned a particular value. That was a negative value the rat learned. What's happening psychologically is this. This is Bindra-Tote's control, the rules of incentive salience. This is how incentive salience works. Pavlovian cues take on motivational value associated with reward, but they don't just take it on in a learned sense. Just as physiological states can modulate the value of an actual unconditioned reward like Dead Sea saltiness, that's called alliesthesia, a term invented by Michel Cabanec at Laval University. The pleasure of saltiness changes with our physiological state. Just so, physiological states can directly change the motivational value of their Pavlovian cues, a sort of conditioned Pavlovian alliesthesia. You don't need to retaste and have new read learning. The alliesthesia extends to the cue. But it only extends when the cue is present. People have been given salt appetite. They wake up and they're like Tony Dickinson's rats. They don't know what they want. They don't necessarily say salt. They don't say that. Some of them just say, I feel a little queasy. But present a Pavlovian cue, and Tony Dickinson has done the experiments with rats where the rats behave, they need experience, they need retasting. But if you present a cue, a Pavlovian cue, you get this while the cue is present, and vivid imagery too. It's adaptive here, but it can be the same kind of thing. This is a Bindra-Tote's diagram. The original diagram was drawn by Fred Totes, who sort of mapped this physiological control of wanting on the cues in the diagram. And Terry Robinson and I just split this diagram into wanting versus liking to say it's happening simultaneously in both to create this kind of changes in wanting without having to relearn. It's adaptive in that case, but that same kind of feature, remember the features, they're pulses of wanting triggered as momentary peaks by the cues. The Pavlovian cues are the triggers. Those cues become attractive, wanted motivational magnets themselves, as well as triggering wanting for the rewards, but their power is utterly manipulable by current physiological and brain states. You just saw it modulated by a physiological appetite state, but all kinds of mesolimbic brain states are going to modulate it too. So here we'll see a few modulations. Things like dopamine activation. Here we're going to reach right into the nucleus accumbens and just spritz amphetamine into the nucleus accumbens in a microinjection. And the rats are going to be working along in extinction for a reward in something called Pavlovian instrumental transfer. What they're doing here is they're working in extinction for sugar cues. They're pressing a lever for sugar cues. And every once in a while, a little Pavlovian cue will come that means sugar. Beep, a sound that means sugar. The rats have never heard this sound while they're working before. They had always learned it separately. But when they hear the sound while they're working in extinction, and the curve is going down because they're in extinction, no sugar is actually coming, every time they hear the sound, there's a little peak of wanting. From wherever they were before the sound came, there's a little peak, a rise up, a little rise up. What happens if you give dopamine? Do they just massively seek all the time? No, they don't. What we're seeing here are the effects of either vehicle or amphetamine put into the shell or core of the nucleus accumbens on different aspects of this trial. If you give amphetamine, the baselines, these little white dots, never go up. The rats don't work harder in the absence of a cue. In extinction, they don't work harder, even though their brain is flooded, their nucleus accumbens is flooded with dopamine. But when the cue comes, the peak of the mountain rises, even though the plateau doesn't change at all. Just the mountain peaks rise up, the cue-triggered mountain peaks. It's an amplification of cue-triggered desire. It's not a general desire or a seeking or activation or a behavioral. It's a cue-triggered motivation interacting with brain state, just like in the salt case. Amphetamine does that. Things like CRF, microinjected in the nucleus accumbens, a stress hormone, do exactly the same thing. Stress primes vulnerability to relapse in addiction, to relapse in binge eating. Stress, even a happy stress like winning the lottery or getting your paper published or getting a great grade or getting a promotion, can trigger relapse. It acts in this way, I think, at least in the nucleus accumbens, by amplifying cue-triggered wanting. In these ways, it mimics dopamine. Very last thing. What I've been trying to do here is to peel wanting away from liking and learning, and to some extent we've peeled it. But what I'd really like to do is sort of offer you wanting on a plate, separated really from everything else. And that hasn't been possible to do with the neurobiological manipulation so far, but it's now, I think, maybe becoming possible. So let me show you just an example or two of this. This is tapping into the mesolimbic system in the nucleus, in the amygdala, in the central nucleus of the amygdala that's interacting with the dopamine systems as people here are studying. What we're going to see is an example of wanting without like that really detaches from these other things. We're going to use optogenetic brain stimulation for a rat who has a happy choice. This rat will have a happy choice. It can press a lever and earn a sugar pellet, or it can press another lever and earn exactly the same kind of sugar pellet, an equal sugar pellet, but also get its amygdala stimulated at the same time. The amygdala stimulation will be by laser light stimulating the optogenetic systems. Most of you, I think, have heard of this before. So the rat has received a microinjection of a virus in its nucleus accumbens. The virus carries a gene for photoreceptor molecules. The infected neurons in the amygdala make the photoreceptors and sprout the photoreceptors. So now the light will open sodium and calcium ion gates and make those neurons fire. If we put this together with the rat who has this happy choice, here's what we're going to see. You're going to see first the one lever comes out, and the rat gets it and gets a sugar pellet with its laser. Then you're going to see the other lever come out, and the rat seems very interested in it. It works just as hard and gets its sugar pellet alone. Then the two levers come out simultaneously, and the interesting thing is what happens when the rat has a choice here, when it has a choice. Okay, yeah, so we'll see this focus wanting. So first this lever will come out. The rat nibbles on it, pushes it, has to push it 8 times. It got the sugar pellet. Here's the sugar pellet in this dish there, and it's getting the laser. There's a little timeout period. Okay, it got that sugar and laser. Now this other lever pops out. It seems just as interested, very, very eager, nibbling on it, nibbling, pushing it, earns the sugar pellet, gets another cue, got the sugar pellet there, so it seems equally interested in both. What happens if it gets a choice? Well, it went to the laser lever and got a sugar pellet. It's gone back to the laser lever. It could earn more sugars by going back and forth, but it's sort of ignoring the other sugar, staying kind of fixated. There's an 8-second timeout period. There it's passed it and it's earned another sugar and laser. It seems very focused. Give it one more chance. One more chance. That time it looked at the sugar lever, but it just went back. And this is what it does. Once it's acquired this, it stays sort of obsessively focused on just this one target and ignores the other, even though one at a time. A thing in addiction has been, it's not just that we want things intensely, it's that we want one thing in intensely. It becomes very, very focused. What's controlling this? Well, the amygdala might be participating, but why is it doing it psychologically? It's happening here wherever the central nucleus of the amygdala is being stimulated. why does this rat only want this laser and sugar when it has the choice? Is it wanting the sugar because it's the laser somehow making this sugar better, making it like the sugar more? Is it wanting this because it just wants the laser alone? Is that what it wants? We could ask the rat. Let's ask the rat. Does it want the sugar? Does it because it likes the sugar more? We could do the taste reactivity test with the sugar in the presence of the laser and in the absence of the laser, and the answer is there's no change. The rat does not like the sugar more. It doesn't want the sugar more because it likes the sugar more during the laser. There's just no effect of laser on liking. Okay, well then it must want the laser more, right? So we can ask the rat, does it want the laser, by saying you can touch, here's two spouts, two things in the world. You can touch this and get a laser pulse, or you can touch this and get nothing, and just let it touch. And what you're seeing is the rat samples a few times at both, but doesn't really touch any of them very much. It's not touching the spout anymore. Or we could go back to the original brain stimulation reward discovery by Jim Olds and Peter Milner. What Jim Olds did in the classic discovery you might have read is he put a rat on that very first day on a table, and whenever the rat went to a particular corner, he would stimulate the electrode in the lateral hypothalamus, and by the end of the session, the rat was all in this one corner. So it wanted to stay in the corner where it was getting the electrode stimulation. Let's try that with this rat, and we can put the rat in a chamber where it can wander about. The periphery of this chamber, there's a big cylinder in the middle that blocks the center, so it just wanders about the periphery. And the size of these circles tells you the percentage of time it spends in the four corners. This circle has the laser, and basically it's just 26%, 25%, 24%, 24%. There's really no difference. It doesn't seek out the laser corner. It doesn't avoid the laser corner. It doesn't care about the laser corner. It's just absolutely neutral. This is the same rat that was pursuing the sucrose. Paste this laser on a sugar, and it becomes an intensely desirable sugar. Paste this laser on something else, like this or this, and it's nothing. It's not just sugars. You could paste this laser on something else, like cocaine. How about cocaine? Shelley Warlow has done the same experiment with cocaine, intravenous cocaine. We'll go back just 1 second. This rat will have a similar situation. It can poke in one hole and get intravenous cocaine, painlessly delivered through an intravenous cannula that's connected, an intrajugular cannula that's now connected to a delivery tube while it's in this chamber. Painless delivery when it pokes its hole in this thing. One just gives it the cocaine, another nose poke hole will give it cocaine plus laser. So let's see what happens. So this is the regular cocaine, no laser. It just poked, and now it's getting cocaine. Now here's another nose poke. It pokes, and it's getting cocaine plus the laser. Seems quite eager. Give it a choice. It looks but doesn't trigger yet. It looks but doesn't trigger yet. It looks and triggers the cocaine with the laser, and it's going to now stay on this porthole all throughout the trial. Just like it did for the sugar. You see, it'll be focused on that one cocaine just like before. Again, give this rat the chance to earn laser alone. Nothing, even though it makes it want cocaine. It's not just making it pursue this cocaine, it's also taking on incentive salience qualities. Remember the incentive salience features? It makes a cue not only trigger motivation, but also attractive. Cocaine cues are different from sugar cues, if anybody's done those studies, and some of you have. A sugar cue you've seen, a rat nibbles on. A cocaine cue, rats don't nibble on. They approach, they sniff, they kind of go back and forth, almost an approach avoidance kind of thing. They don't nibble on a cocaine cue. It's not in the literature. But paste a laser on a cocaine cue and add cocaine in the course of the trial, and what we're going to see is that this cocaine cue becomes especially attractive in a whole new way. Oh, I should have mentioned, if you take rats and you want to make them, if you give them a choice between sugar versus cocaine, paste the laser on the sugar and they become a sugar addict. Paste the laser on cocaine and they become a cocaine addict. You can make them pursue whichever you want. But let's see what they do to the cue. Here's a cocaine cue for this rat. This is cocaine with the laser. And here it's at the end of the trial doing something that I don't think has been observed before in cocaine studies, of rats actually trying to gnaw and eat, in a sense, there we go, to bite this cocaine cue as though it were a sugary cue. This cue is becoming fascinatingly attractive in a way that cocaine cues, even cocaine cues, normally don't actually get. This is a different rat. She's in stereotypy because she has lots of cocaine on board, but she's still trying, she's still trying. Give her one last chance. What will she do? Let's see it in slow motion. Not very good bites, not very good, not very good, not very good. A little more chance. There we go, that actually moved. She pulled that out, so that was a very good, serious bite. That's what it does to that kind of cocaine cue. Very last thing, last 30 seconds. We know what happens if you paste a laser on a sensory reward. It becomes this obsessive focus of desire. So what if you were to paste this laser upon a nasty event, a nasty object? Pinnell and Treat here at the UBC, I think, began the shock prod natural defensive burying paradigm, where if the rat touches a little prod, it gets a shock, doesn't have to touch again, so it can bury and defensively bury and sort of bury that object. So this rat has buried it. This is from a Pinnell and Treat kind of study. Let's paste the laser on the shock prod. First, let's see a normal rat with the shock prod. Then let's see a rat who has the laser pasted in the shock prod. That's a normal rat. Got one shock. Whoops, shock. Stay away. Stay away. Defensively. Bury a little bit. There you go. Starting that. Here's a laser rat. Oh, shock. Oh. 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 That's, oh. Oh. That's the difference. She gets the shock. She knows she's getting the shock, but there's something fascinating about this shock. Now, what is going on here? This is something that we don't really have a good psychological sort of toolbox for explaining. It's not a reinforcement history. It's not a collection of reward associations. It's sort of taking the desire and sort of pasting it in the presence of this nastiness on this nasty object. Is it related to things like self-harming? Is it related to things like masochism? Who knows? But what it's showing us is that brain systems of desire can be pulled apart from the usual kinds of psychological explanations we have for desire. They are active systems, and they may be involved not only in daily life, but potentially in certain kinds of psychopathologies. So that ends me, really. The whole theme of today has just been to say we can break reward apart psychologically, and to some extent map it onto particular brain systems, at least pull them apart through brain systems. Wanting, liking, and learning, they usually go together in daily life. They're usually together. But dissociations do happen. In the lab, we've seen them, and they may happen in daily life, too. We shouldn't assume that they're all together. You can have wanting without liking, potentially an addiction if sensitized addicts want drugs that they don't particularly like. Wanting without liking for the laser prod. You can have liking and learning without having any wanting for what you've learned to like, which may happen in dopamine loss, and possibly in clinical avolition syndromes that used to be thought of as anhedonia syndromes. People might like and still be able to learn and yet not want the things they're learning about that they've liked. You can have wanting and liking without learning, as in the Dead Sea Salt cue, where you wanted and liked something that you had never learned a positive reward association to. And finally, psychologically and neurally, even though wanting seems totally different from fearing or dreading something, there is something overlapping in the neural systems and potentially in some psychological features too, like this motivational salience of things and the recruitment of active sort of dopamine kind of mediated responses. The nucleus accumbens, we can combine desire and dread, and here in the amygdala laser X thing, you're seeing this irrational desire for something which is active, which is becoming a target and becoming eligible for desire because it's delivering a nasty event. Place the laser on a neutral thing, and there'll be no desire for the neutral thing. So I should stop there and simply say that I haven't done any of this at all. It's all been done by people who are more talented than me, and they are all listed here. And I'm very grateful to them, and thank you for listening.
Through his lunch hour. He has not eaten lunch yet, but we have time for a few questions before we feed him.
So with respect to the difference between the Berlin-Dickinson incentive shifts, where the animal has to retaste, and the Dead Sea Salt kind of experiment, and the fact that there's not a Pavlovian cue in the Berlin-Dickinson experiments, whereas there is one in the sea salt experiments, it's just interesting to me because the levers do come out in the Balayan Dickinson. There is a lever that the animal has learned. If you respond on this, you will get a sugar pod. So is it because it was never like framed as a discrete cue that you get this instant relearning, or what do you think that is?
Yes, I think it's because Tony Dickinson is a master at this. It's true, there are cues that are always embedded in things, but Tony takes special care to assure that every cue has an association with every reward coming in the same places. So it's not that they aren't cues, it's that they have Pavlovian associations. He has them in two choice situations too, where only one cue's value is changed, one reward's value is changed. But the cues that are present have associations with both values. So sometimes there's a change in overall responsive, but it's never specific in direction for the wanted one. So he's actually done this salt appetite experiment. He has done it twice. 1986 was the last one. And he gets no change in the response when the rat wakes up in the salt appetite until it gets a taste of the saltiness. It was a thing for salt versus water. Now, he is such a master that there are people who do Tony Dickinson experiments who can't replicate some of his effects, like Bob Rescorla, who has done some of the same salt experiments and will get instant shifts. Tony explains that as Bob not having the same kind of control over the Pavlovian cue. But everybody agrees, I think, that if you provide a Pavlovian cue, and Tony in separate experiments has done it, where a Pavlovian cue happens, you get dramatic changes in motivation. Now, in his case, the cue is a sound. It doesn't tell you where to go. And when he gives the cue for salty water, a sound, the rat goes frantically towards both reward options. One is salt and one is just plain water. Only one makes sense at this moment. So he says it's not directional. But allow a cue to be a bindra cue, where the cue can be a beacon, directional beacon, as well as an informational trigger. And that becomes attractive and wanted in itself. And that's what I think pulls these in. So I think they're both true. They're just parallel psychological processes, and manipulate the situation, you can expose them. Luke?
Great talk, thank you. So on opioids and liking, you show very clearly that those opioid-liking effects are very, very localized in the brain to this small number of hotspots. I'm interested in what you feel the clinical implications of that are, because in human diseases we're generally giving drugs systemically or orally. So how much of an impact does a drug like naltrexone have on liking responses when the brain basis is so localized like that? And can you look at that in the animal models? Right.
Well, I think opioid manipulations change both wanting and liking. So we can, with opioid in the hotspot, we get liking and wanting, but opioid out of the hotspot and the rest of the nucleus accumbens, we get wanting without any liking. If we do something like in a clinical situation, like an addiction, and give naloxone, we might well suppress the liking, but we also might directly suppress or modulate the wanting. So I think many clinical manipulations will be potentially affecting both, because it can behave as both. In terms of implications of the localization, I think, in a sense, it's like 1990 all over again. We have this in the rat, like then we had the dopamine being wanting in the rat, but there wasn't any human thing we could point to say that dopamine was only wanting and not liking in people. Everybody knew dopamine was liking in people, or Volkov and many others. What we can say is that when pleasures can be pinned in human brains, I'm hoping that these localized hotspots will turn out to be important. There have been a couple studies, like Andy Calder before he died. He had a study where he was showing people delicious foods or disgusting rotten foods. And in the ventral pallidum, our hotspot is in the posterior part. Opioid turns on liking and wanting. Move the opioid stimulation to the anterior ventral pallidum, and we get a suppression of liking and wanting. It's like a seesaw. Yeah, I think he was finding in that study they reported posterior ventral pallidal activation to the delicious foods, anterior ventral pallidal activation to the disgusting foods. It's rare to get this kind of claim for that kind of anatomical localization, and that, but that's the kind of thing that I would look for first. And then for clinical syndromes, I don't have a clear prediction, but perhaps something will pop. Yes?
The case where you said there was a liking and learning, but no wanting. What was that case? case, did taste divergent case? Was it?
So the liking and learning without wanting. So in the laboratory, that might be a rat who's had a big dopamine lesion and who seems to like things normally and can learn new likes and new dislikes, but who doesn't want anything. Pardon?
What kind of learning? Is it?
Well, that was a taste aversion learning.
Oh, I see.
But people like, say, Richard Palmer using dopamine-free mutant mice can have them learn locations of rewards and things like that without dopamine too. A human case might be, but less extreme, would be if it is true that anhedonic Parkinson's patients and anhedonic schizophrenics and depressives who turn out to actually have normal pleasure ratings, when you give them, taste this ice cream and tell me how do you like it, oh, it's normal. But nothing in their life has value to them in the same sense that it once did. So that would be, people like Deanna Barsh and Michael Treadway and David Zalt are suggesting that that's not anhedonia, it's really avolition. They can learn, they have pleasure, apparently, but things are not incentively motivating to them. So that would be.
So the dopamine doesn't increment learning. Is that the
That's a big argument about that. My own answer is I don't think it does. I think it colors what we learn in what we learn normally about them.
That was great. Thank you so much. So I don't really know anything about neuroscience, so this question could be totally off. But I was just trying to sort of figure out some of this rat stuff in terms of how it explains human behavior and human approach and avoidance behavior in particular. And the last thing you presented was really kind of fascinating, obviously, where you have the rat clearly wanting the thing that's causing presumably pain. And I'm wondering whether, do you think that could parallel kinds of stuff we see in humans when they go after thrill-seeking type things that are going to be, like, I don't know, I'm not a thrill seeker myself, but a lot of humans go after things that I think are intrinsically painful or threatening in some way and have a high drive for that, and I'm wondering whether that's possible.
Yeah. I definitely think that these systems can explain things going after ambivalent things like roller coasters and things that can be good and bad. A scary movie, good and bad, is kind of mixed. That's like the desire and dread rat mixture that was flipping back and forth. What I am delighted to see but wouldn't have hoped for is that it doesn't have to be ambivalent. It can be clearly nasty and still become a target of desire, like the shock pod. And that, I think, is what it's telling me is that these psychological systems of desire can be detached. They have their own life, they have their own rules, they have their own mechanisms. So they can be detached from all the usual explanatory things that we usually have powering them. And this is a bizarre lab experiment, but it's conceivable that some sort of detachment does happen in a person who desires something that's unambiguously bad. And I don't have I'm not claiming that's the case. I'm just saying this is a possibility we should think about.
I mean, I was even wondering, sort of thinking clinical terms about people who are like borderline personality or something, where people are seeking out relationships that are causing pain or something.
Yeah. I think that's conceivable. I had a colleague who saw that and said, oh, it's masochism, basis for masochism, or self-harming where a person seeks out something that becomes damaging. And I wouldn't say that this is the case, but I would say this is a possibility that might be the case.
But on that point, instead of putting this into liking, what about vigilance?
What about?
What about vigilance? Since the fraud is a threat, and you're tapping into the central amygdala, then the animal wants to attack it or pursue it or figure it out.
I think that is happening. That is probably a legitimate way of characterizing this. We want to psychologically, though, ask, why is this the case? I mean, imagine an alternative universe. What could have easily happened is we tap this on, and the amygdala, which is involved in fear, as you point out, the laser could have exacerbated and amplified that. Just as it's been amplifying desire for cocaine and sugar, we could have had a rat who not only touches it once and does a little burying it, but touches it once and then does massive burying and stays far and far away. We could have vigilance that keeps us well away and never, say, pressed against this far wall. That could be vigilance too. What we're seeing is vigilance that's making this just, and is actually going to make us get more shocks, because she does get more shocks, that laser rat. She gets 10 times more shocks. I mean, we stopped the trial because, yeah.
Buried in Pinel's work is that animals that get shock prod and then you put them back, the latency to
Yes.
Because if it was about fear, then the latency shouldn't do.
That is right, and but latency could be relevant. There is a third alternative that could have happened. It could have been vigilance, latency to approach and defensively bury, but not actually touch, right? And I think that's what Pinnell would usually have for that
One more
latency.
on that. Did they show a stretch attend approach? Did they bury the ones that got the laser plus the shock from?
No, they are not burying it. They are sort of staying like this. There are precedents in the literature. I mean, you probably wouldn't have many. Ron Melzak, his doctoral study was actually puppies with social deprivation at the University of Maine. And the little puppies, after several weeks of being raised social deprived, were doing things like approaching a hot radiator and sniffing and sizzling. You know, and that kind of approach to these harmful things was with a psychological sledgehammer experiment that one would never do again. But it may be possible to turn on the expression of these things in more delicate ways.
Kent must be hungry right now. He's certainly wanting some food, I'm sure. So let's thank him one more time.
Thank you.